This is an exam question about the kidney and the roles of the nephron. The question says, explain how the composition of fluid through part C, which is the distant convoluted tubule of the nephron, shown in figure 5.1, is altered by the cell type shown in figure 5.2. So 5.1 is just a picture of the nephron. So this is 5.1, and you can see you've got 3 pictures of nephrons. The question is about part C, which is the DCT. It's asking about the role of this cell in altering the composition of the fluid. So this is a brush border cell, and just a reminder that brush border cells have got lots of visible features that help them in the role of selective reabsorption. So we've got microvilli providing a large surface area, rough ER and ribosomes for the production of proteins, lots of mitochondria for the production of ATP. It's got a folded basal membrane down here, which is providing a large surface area, and it's got tight junctions between the cells to stop any large molecules passing through the tissue fluid. So we've got to talk about how that cell changes the composition of the fluid flowing through the nephron. Remember, the fluid has come from the Bowman's capsule, which has carried out ultrafiltration. So in the fluid we've got everything that you would find in blood plasma apart from proteins and red blood cells. These cells therefore need to reabsorb a lot of those constituents of the fluid back into the blood. For example, amino acids, glucose. So the role of the cell then, of this cell, alright, the brush border cell in the PCT, is selective reabsorption. We know, and again look at the cell, we can see that it's got lots of mitochondria, that this selective reabsorption is an active process. So ATP is required. And that ATP is from mitochondria. Right, so how does selective reabsorption take place? Well, this basal membrane has got a sodium pump. And it actively pumps sodium ions out of the cell into the tissue fluid and then into the blood. So we can mention that there's a sodium pump, and that's on the basal membrane. Don't say basement membrane. It's not the basement membrane. Okay, it's the basal membrane. And it's folded, so it's got a large surface area, so we've got many of these sodium pumps. Right, so we've said that the sodium pump is going to actively pump sodium out of the cell. So that's going to cause a low sodium concentration, sodium ion concentration, inside the cell. Okay, so we've now set up a concentration gradient. We've got low sodium concentration inside this cell, but we've got a high sodium concentration in the filtrate. So this is the filtrate that's come from the Bowman's capsule. So the sodium now will move into this cell by facilitated diffusion. So it moves through something called a co-transporter. So you've got co-transporter proteins on the microvilli, and the sodium then will move in down the concentration gradient by facilitated diffusion through a co-transporter protein. So we can, that's the next thing we need to say, is that sodium ions, again, make sure you mention sodium ions, not just sodium, enter the cell from the lumen of the PCT. And that they use a co-transporter protein. So we have the co-transport proteins then on this brush border cell. Sodium ions are moving in from the filtrate, from the lumen and the PCT, and as the sodium moves in, amino acids and glucose will be pulled in by that co-transporter. Co-transporter means that as one substance moves in, one molecule, it pulls in another molecule as well. So we've got using a co-transport protein, and that co-transport protein, as well as transporting sodium ions, will transport amino acids, and then you'll have separate co-transporter proteins that will transport glucose. And all the amino acids and all the glucose will be reabsorbed. Along with this, because you've got a high concentration of vitamins and chloride ions in the filtrate from the ultrafiltration in the Bowman's capsule, they'll move through the cell and then into the tissue fluid as well. So vitamins are reabsorbed, and chloride ions are reabsorbed. Because you've got a water potential difference between the filtrate and the tissue fluid in the blood, there's plasma proteins and white blood cells, so it's got a lower water potential and more negative water potential than the filtrate. Then water is also going to move through this cell, and that's going to be by osmosis. So we can say that water is reabsorbed and that's by osmosis. You must say the process by which the water is being reabsorbed. Right, some urea, about 50%, is reabsorbed, and that's because urea is generally a small molecule, and that will be by diffusion. It'll just diffuse from a high concentration in the lumen of the PCT to a low concentration in the blood because it's being filtered out in the Bowman's capsule. Right, you've got two other nitrogenous wastes that's being filtered out. You've got uric acid and uric acid is not reabsorbed. And then you've got a chemical creatinine, and that is actually secreted into the lumen by the brush border cells. So as the filtrate is passing along and past these brush border cells, and the amino acids are being reabsorbed, glucose is being reabsorbed, sodium ions are being reabsorbed, water is being reabsorbed, 50% of the urea is being reabsorbed. The volume of the filtrate massively decreases because a large percent, about 80% of the water is reabsorbed, but the concentration in the filtrate actually increases of urea because only about 50% of the urea diffuses back into the blood. So you can say that the volume decreases but concentration increases. And that filtrate then will pass on to the next stage of the tubule, which is the DCT, leading into the loop of Henle, where more water is then going to be reabsorbed. So that's explained how the composition of fluid is altered by these brush border cells in the PCT. You need to make sure that you understand the visible features of the brush border cells which enable them to carry out selective reabsorption and the process of selective reabsorption. So it's an active process, ATP from mitochondria, the sodium pump on the basal membrane that lowers, so sodium moves through the sodium pump, that causes a low sodium ion concentration inside the cell. Sodium ions then enter the cell from the lumen, used as a co-transport protein, where you've got proteins that transport along with the sodium ions amino acids and separate ones that transport glucose. Vitamins are reabsorbed, chloride ions are reabsorbed, water is reabsorbed by osmosis, urea is reabsorbed by diffusion, uric acid is not reabsorbed, creatinine is actually secreted into the lumen, so from the blood into the filtrate inside the PCT, and the volume decreases inside the lumen of the PCT, but the concentration increases.